Hello class. Last time we have learned about the structure and functions of bacterial cell membrane. In this lecture, we are going to learn about the cytoplasm of bacterial cell. The cell membrane surrounds a complex solution referred to as cytoplasm or cytoplastic matrix. Cytoplasm is about 80% water and contains primarily proteins, carbohydrates, lipids, inorganic ions, and many low molecular mass compounds. The cytoplasm also holds larger discrete bodies such as chromosomes, ribosomes, granules, and actin strands. First, let's learn the bacterial cytoskeleton. Eukaryotes possess three cytoskeletal elements: actin filaments, microtubules, and intermediate filaments. Actin filaments are made from actin and microtubules are made from tubulin. Intermediate filaments are composed of a mixture of one or more members of different classes of proteins. As the table shows, homologous of all three types of eukaryotic proteins have been identified in bacteria. The bacterial cytoskeletal proteins are structurally similar to their eukaryotic counterparts and carry out similar functions. They participate in cell division, localize proteins to certain sites in cell, and determine cell shape. The best studied bacterial cytoskeletal proteins are FITZ, MARB, and CRAS. FITZ was one of the first bacterial cytoskeletal proteins identified and has since been found in most bacteria. FITZ is a homologue of the eukaryotic protein tubulin. It forms a ring at the center of a dividing cell and is required for the formation of the spectrum that will separate the daughter cells. MARB and its relative MBL are actin homologous. Their major function is to determine cell shape in rod-shaped cells. MARB is not found in cocci. MARB and MBL determine cell shape by properly possessing the messenger needed for peptidoglycan synthesis. CRAS is responsible for its curved shape. CRAS is a homologue of lamin and keratin, two intermediate filament proteins. Next, let's move on to the intracytoplasmic membranes. Although members of bacteria do not contain complex membranous organelles like mitochondria or chloroplast, internal membranous structures are observed in some bacteria. This can be extensive and complex in photosynthetic bacteria and in bacteria with very high respiratory activity, such as the nitrophilin bacteria. The internal membranes of the photosynthetic cyanobacteria are called thylakoids and are analogous to the thylakoids of chloroplasts. Inclusions are common in all cells. They are formed by the aggregation of substances that may be either organic or inorganic. Inclusions can take the form of granules, crystals, or globules. Some are amorphous. Some inclusions lie free in cytoplasms. Other inclusions are enclosed by a cell that is single-layered and may consist of proteins or of both proteins and phospholipids. Some inclusions are surrounded by invaginations of the plasma membrane. Many inclusions are used for storage or to reduce osmotic pressure by tying up molecules in particulate form. The quantity of inclusions used for storage varies with the nutritional status of the cell. Cells have a wide variety of storage inclusions. Many are formed when one nutrient is in ready supply but another nutrient is not. Some store end products of metabolic process. In some cases, these end products are used by the microbe when it is in different environmental conditions. The most common storage inclusions are glycogen inclusions, PHA granules, sulfur globules, and polyphosphate granules. Some storage inclusions are observed only in certain organisms, such as the cyanophycin granules in cyanobacteria. Carbon is often stored as polyhydroxyyakunate granules. Several types of PHA granules have been identified, but the most common contain poly-beta-hydroxybutyrate. The structure of PHB inclusions has been well studied, and PHB granules are now known to be surrounded by a single-layered cell composed of proteins and a small amount of phospholipids. Much of the interest in PHB and other PHA granules is due to their industrial use in making biodegradable plastics. Metachromatic granules are large inclusions that take their name from the fact that they sometimes stain red with certain blue dyes, such as methylene blue. Collectively, they are known as volutin. Volutin represents a reserve of inorganic phosphate that can be used in the synthesis of ATP. It is generally formed by cells that grow in phosphate-rich environments. Metachromatic granules are found in algae, fungi, and protozoa as well as in bacteria. These granules are characteristic of diagnostic significance of Cronibacterium cereus and Mycobacterium tuberculosis. Polyphosphate granules store phosphate needed for synthesis of important cell constituents such as nucleic acids. In some cells, they act as an energy reserve, and the polyphosphate also can serve as an energy source in some reactions. Sulfur globules are formed by bacteria that use reduced sulfur-containing compounds as a source of electrons during an energy-consuming metabolic process. For example, some photosynthetic bacteria can use hydrogen sulfate as an electron donor and accumulate the resulting sulfur either externally or internally. Carboxysomes are inclusions that contain enzyme ribulose 1,5-bisphosphate carboxylase. Photosynthetic bacteria use carbon dioxide as their sole source of carbon and require this enzyme for carbon dioxide fixation. Among the bacteria containing carboxysomes are nitrifying bacteria, cyanobacteria, and acidicylbacilla. Magnetosomes are intracellular chains of magnetite or graphite particles. The magnetotactic bacteria use magnetosomes to orient themselves in magnetic field. They are around 35 to 125 nanometer in diameter and enclosed with imaginations of the plasma membrane. For a cell to move properly within a magnetic field, magnetosomes must be arranged in a chain. Hollow cavities found in many aquatic prokaryotes, including cyanobacteria, anoxygenic photosynthetic bacteria, and hollow bacteria, are called gas vacuoles. Each vacuole consists of rows of several individual gas vesicles, which are hollow cylinders covered by protein. Gas vacuoles mating buoyancy so that the cells can remain at the depth in the water appropriate for them to receive sufficient amounts of oxygen, light, and nutrients. Ribosomes are the site of protein synthesis, and large numbers of them are found in nearly all cells. The cytoplasm of bacterial cells is often packed with ribosomes, and other ribosomes may be loosely attached to the plasma membrane. Bacterial ribosomes are called 70S ribosomes and are constructed of a 50S and a 30S subunit. Bacterial ribosomes are composed primarily of ribosome RNA molecules. The small subunit contains 16S rRNA, 23S and 5S rRNA molecules are in the large subunit. Approximately 55 proteins make up the rest of the mass of the ribosome, 21 in the small subunit, and 34 in the large subunit. The nucleoid is an irregularly shaped region that contains the cell's chromosome, and numerous proteins. For most bacteria, the nucleoid is simply a region in the cytoplasm. It is not separated from other components of the cytoplasm by a membrane. The chromosomes of most bacteria are single-circle double-strand DNA, but some bacteria have a linear chromosome, and some bacteria such as Vibrio chloride and Borrelia burgdorferi have more than one chromosome. Bacterial chromosomes are longer than the length of the cell. How these microbes manage to fit their chromosomes into relatively small space occupied by the nucleoid. For instance, E. coli's circular chromosomes measures approximately 1,400 microns, or about 230 to 700 times longer than the cell. Thus, the chromosomes must be organized and packaged in a manner that decreases its overall size. Supercoiling is thought to be important. It produces a dense central core of DNA with loops of DNA extending out from the core. Several nucleoid-associated proteins cause the chromosome to bend and fold, thereby also helping to pack the DNA into smaller space. Okay, that's all for today's lesson.